Okay, so last time we left off on chitin. So chitin is going to be the main structural polysaccharide of things like fungi and arthropods. This being an arthropod right here, right? That being a chitin shell. So chitin is going to be the homopolymer of beta-1,4 linked N-acetyl glucose amine. Notice right here, there's the amine, the acetyl, there's your glucose, right? Okay, and again, that's that fibrous structure. It's found in fungal cell walls and exoskeletons and shells of arthropods. The fibers are often cross-linked and composited with minerals and proteins to increase the rigidity and strength. And so you can incorporate things like calcium oxides in there to give them more rigidity, and you can do a lot of work with them. So this is sort of like the, you ever had like Kevlar plates? Ever seen those? Okay, like that. Kevlar is not a plate, right? Not normally, right? Very similar. Also, the wings of insects, chitin. Okay, so the chitin can be processed to a molecule with a variety of uses. The cellulose is a major component of paper, bioadhesives, and clothes. Chitin can be recovered from shellfish industry by processing the shells into more versatile chitosan through microbial and enzymatic processes. The chitosan then can be used as a carrier to assist in drug delivery, component of cosmetic and food products, surgical dressing. There's actually a really high-priced glue that's made out of chitosan. It's very, very good glue. It's really strong for its amount that you'd use. Actually, there's a Japanese manufacturer of chalk whose chalk boxes spiked up to hundreds of dollars per box because they're going out of business, and they actually grind down shellfish shells and put it in to give it more structural rigidity so it doesn't break like normal chalk, right? Yeah. So the carbohydrates, these can be linked to proteins to form glycoproteins. Glycoprotein is exactly like it sounds. It's going to be a carbohydrate group covalently attached to a protein. Makes up about 50% of the human proteome. Glycosylation, this is going to be the increase of the complexity of the proteasome. The glycoforms are going to be different Let me get my water real quick. Oh, man. Different glycosylated, Okay. These may occur when a protein has several different potential glycosylation sites. This is not my day for this. I really need like a singing practice before this. Maybe join the choir or something, right? Anyways, you can have multiple sites to glycosylate to form different glycoforms. The three classes of glycoproteins. So you have glycoproteins, which are predominantly proteins. These are going to play a variety of roles. This will include things like cell adhesion. Proteoglycans, these will be predominantly carbohydrate. And the protein component is going to be conjugated to the glycosaminoglycan. The function as structural components and lubricants typically. Mucins, mucoproteins being the full name of it, by the way. I personally think the mucoprotein is easier to do. Mucins is kind of a weird name. Predominantly carbohydrates, and the protein components is extensively glycosylated at the SER or 3 residues. Usually by N-acetyl galactosamine. Notice these two are kind of similar, but they have an important difference, right? Very important structural difference. Okay, this is actually your key component of mucus, as you probably figured from the name, right? Mucoproteins. Mucus. They function typically mostly as lubricants, which is what your mucus largely functions as. The carbohydrates can be linked to proteins through the N-linked or O-linked. Again, the N-linked is going to be bonded to the amine, the nitrogen. The O-linked will be bonded to an oxygen. Okay? So your N-linkage is going to link the sugars and the glycoproteins to the amide nitrogen atom, typically in the side chain of ASN. That's this one right here, by the way, in case you forgot. The ASN, this has to be part of an ASN-X, that's right here, SER or an ASN-X3 sequence where the X is going to be any other residue except for proline. Again, this would be like, actually it's basically like we're seeing right here. Where you have this gap right here is something else, typically something like proline. God, that figure actually worked out very nicely, didn't it? Nicer than I even realized. So the O linkage, this is going to link the sugar and the glycoproteins to an oxygen atom, typically in the side chain of SER or 3. And this is going to be like this right here. You probably actually figured this out from this right here is an amide linkage. Can everybody see it okay? And see that that was almost an amide group in the first place, right? So it's fairly easy to do that linkage. This one right here is just your normal oxygen ether linkage that we've been looking at, right? Okay. Everybody's clear on why that's O and N and how that works out, right? Okay, fantastic. Okay, so the N-linked oligosaccharides, these have a common core to them. The N-linked polysaccharides have a common pentasaccharide core. This is going to consist of 3 mannoses and 2 N-acetyl glucosamine residues. And these are the common cores right here. See the boxes right here are the N-acetyl glucosamines. Everybody see that box is okay? And then the green circles here are also part of the common core, which is the mannoses right here, the 3 mannoses. Okay? Why is this one not part of the common core? Yeah, it's not part of any common core, is it? It's not uncommon. Okay? Also note that this is not part of the common core either. Please note that these are linked to ASN here in both cases. Does anybody have any questions about that common core? Okay. Okay, very good. The glyco... Did somebody mention something? Oh, I thought I heard a question. I guess I'm hearing. Okay. The glycoprotein erythropoietin is a vital hormone. The erythropoietin, EPO, that's so much easier, is a glycoprotein secreted by the kidneys into the blood serum to stimulate production of our red blood cells. Their cloned recombinant form has a improved treatment for anemia, but has been abused by some endurance athletes because, of course, right? Anything that increases red blood cells is going to be abused by any athlete. Glycosylation will enhance the stability of the protein in the blood, which is why it can be abused and why it's a more effective treatment, right? Is if you can give a more stable version, which will keep it in the blood and active more often, then that will be a better treatment, but also more effective than your normal. The oligosaccharides is going to be attached to the erythropoietin. This is right here. So the N-glycosylated at 3 ASN residues. Please note, literally, this is what we were looking at over here, right? We've just attached them to the actual protein here. Okay. This is going to have an O-glycosylated serine residue. 40% of the carbohydrate by weight. And so the weight here of these carbohydrates is 40%. Okay? Everybody see it okay and understand what they're looking at? Fantastic. If you want to know the exact identities of each of these things, this is the key over here. Okay? These are actually quite repetitive. They usually use the same shading and color on almost every figure if you're going to do it for a manufacturer. Or like a book, in this case. They're very consistent. So the glycosylation functions and nutrient sensing. By the way, that's why I say you should buy this book at some point in your life, is because it's just a really well done book. Yeah. Anyways, functions and nutrient sensing, your glycan acylation, the post-translational covalent attachment of a single N-acetyl glucosamine. This is where the abbreviation comes from. Can everybody see that abbreviation? How that'd be related? Okay. So glucose in acylation, right? Okay. This is going to be attached to a ser or 3 residues of your protein. It will be catalyzed by, I'm sure you're not surprised, substrate's name, O-glycan-as. Transferase, because we're transferring it over onto there, right? So transferase. This is going to be occurring when the nutrients are going to be abundant. This is reversible, so you can chop these back off. This is actually going to be chapter 16, where we really start talking about that, where we're talking about glucose signals. This is going to tell us that we have carbohydrate that's available and tell us how much is available approximately. Okay. By the way, this is the nitrogen signal right here for protein availability, which we'll get into in chapter 23. And this is the acetate signal for fatty acid availability, which will be chapter 22. So I know, don't let it overwhelm you. That's why we only we'll talk about these things later. Okay? Just concentrate on this overall structure for now. Did anybody see your web figure in your book? It's your metabolic web, and it has all the different chapters color-coded, and they all are unpacked on top of each other. I forget if it's in the front or back, but when you're done with all this, look at it, and you can really appreciate what you've learned and how confusing it would have been to just look at it all at once. Okay. Anyways, this right here is that GLC-NAC right here that's going to be attached, okay? Anybody have any questions about that so far? Okay, so let's talk about the transferase now. The glucan acylation sites, or GLC-N acylation sites, these are also potential phosphorylation sites. So our which one makes more sense to you guys, GLC-N or glucan? Okay. As long as we understand each other, either way works. Okay. If we don't understand each other, then it doesn't work. All right. So again, this is that enzyme, the transferase here, and protein kinases may be involved in crosstalk in this process here, because they both work on that same site. Improper regulation of our O-glucan-as transferase has been linked to these various different, some of these are diseases, some of these are effects or features, as it were. But insulin resistance, diabetes, cancer, neurological pathologies. There's a hugely interesting new body of research that looks at various different neurological diseases, things like Alzheimer's, things like bipolar, and is starting to show that these are actually metabolic diseases. Yeah, they're effects of metabolic diseases. Anyways, yeah, it's some crazy stuff's going on lately. Really cool, but really kind of really interesting, but really confusing. Anyways, proteoglycans, these have an important structural roles, or maybe not confusing in the long term, but right now, completely rewriting all of our medical knowledge. Okay. Proteoglycans, these are going to be up to 95% glucose aminoglycan by weight. This is going to resemble a polysaccharide more than a protein. Proteoglycans are going to function as lubricants and structural components in connective tissue. They'll mediate adhesion of cells to the extracellular matrix, in case you ever wondered what the glue was there. There we go. This will have bind factors that regular cell proliferate. Okay, anyone want to say that for me while I drink? Thank you. Good God. I need some of that spray stuff, don't I? Man. Bind factors that regular cell proliferation. Okay, so glucose aminoglycans, again, is what we're concentrating on. The glucose aminoglycans are going to composed of repeating units of disaccharides containing derivative of an amino sugar. The amino sugar derivative, I want to say derivation, is either the glucosamine or the galactosamine. At least one of the two sugars in the unit has a negatively charged carboxylate or sulfate group. The inability to degrade glucosaminoglycans will cause diseases marked by skeletal deformities and reduced life expectancies, in case you're wondering. These are different repeated disaccharides. Remember, the disaccharides are repeated units that are going to be involved in this. These are them right there. So again, these are those repeating units you'll see. Notice the sulfate here. Sulfate being one of the most common ones, markers of this. This is heparin, by the way, in case you ever wondered what the hell heparin was. Okay. I don't think the other ones are quite as famous. Anybody ever heard of the other ones? Okay, you've all probably heard of heparin because of like medical shows, right? Yeah. Anyways, proteoglycans, these are going to be important components of cartilage. Cartilage will contain collagen protein, and the proteoglycan aggrecan. Aggrecan is actually the large molecule with 3 globular domains. The site of the glucose aminoglycan, in this case, keratin sulfate and chondroitin sulfate, which were up here, keratin sulfate and chondroitin 6 sulfate right here. Okay. These are going to attachment is going to be the extended region between G2 and G3. The G1 non-covalently binds to a central polymer of hyaluronate. Excuse me. This being the hyaluronate. Okay. So the proteoglycans from cartilage is going to have an enormous and complex structure. We can actually see this to some degree right here. This would be your overall large structure. See how spread out and large that is. And then this would actually be what it looks like if it was, labeled for you. Okay. And here's that G1, G2, and G3 that we were talking about before. That's those. Okay. And so right here we have the chondroitin sulfate right there. This is the keratin sulfate. This right here is the hyaluronate. This is the aggrecan in total. Okay. Can everybody see those okay? Okay? Makes more sense when I sum it up into a diagram, doesn't it? Yeah. Okay, so the aggrecan is going to cushion compressive forces. Water is going to be bound to the glucose aminoglycans. This will help to cushion compressive forces. Water is squeezed from the glucose aminoglycan under pressure. Water then rebinds when pressure is released. Essentially, same thing as, anybody ever seen like a waterbed or one of those water cushions? Very similar, right? Just a water cushion, essentially. And so you break up the water, it absorbs force, and then you release it, it goes back. So osteoarthritis, this is going to be a form of arthritis that results when water is lost from the proteoglycan with aging, in which case it starts becoming brittle and it can't absorb forces, right? And so you'll begin to have fracturing, and it will eventually wear down, resulting in loss of your cartilage structures around your like discs and bones and stuff. Okay, where were we? Right here. Okay, so the mucins, these are going to be glycoprotein components of your mucus. Sounds like a few people have some mucus going on. Yep. Okay, so you are fully experiencing these right now. All right, appreciate. Your tandem repeats, VNTR region, the region of the protein backbone of mucins that's going to be rich in the O-glycosylated serin-3 residues. Your core carbohydrate structures are going to be conjugated to your protein component of your mucin. And so this right here, again, is this is the cis region, rich region right here. This right here is the total VNTR region. And again, these are going to be O-glycans right here, because that's the region that's rich in O-glycosylated serin-3 residues. This right here is your D domain, by the way. And here you can see a different representation where it tells you each individual one, but I actually think you lose a lot being able to see this. This again is, the serin-3 residues right here. This is what's off of it. Okay, these are these little chains you see, the squigglies. Yeah, everybody get that? Okay. I know it summed up. It makes kind of a weird picture in your head, right? You can see that these are the little chains, right, coming off here. Okay. So the functions of your mucins, they're going to adhere to epithelial cells. They'll act as a protective barrier, basically a lubricant against being rubbed on and messed with. They'll hydrate the underlying cells. They're going to play roles in things like fertilization, the immune response, and cell adhesion. You all understand what that just said, right? Or do I have to explain bird and bees? No? Okay. Fantastic. Please do note that any of the portions that are not going to have layers of dead cells build up on them, your skin is layers of dead cells. Okay? And so they don't need to be hydrated because the layers of dead cells keep them hydrated, right? You're not actually exposing your live cells. But your live cell areas like your throat, your mouth, your lungs, all of those need mucus, right? Otherwise, they're going to lose water too fast, and you'll end up dehydrating your cells and killing them, won't you? Okay? Or another way of saying that is they'll no longer have flexibility. Because remember what happens when you dehydrate your cell structures. They become brittle, right? Okay. Okay. Did that make sense to everybody? Fantastic. So overexpression occurs in bronchitis, cystic fibrosis, and adenocarcinomas. The protein glycosylation is going to take place in your lumen of your endoplasmic reticulum and in your Golgi complex. Again, this is endoplasmic reticulum right here, Golgi complex. Has everybody seen these before? Everybody's had a class for, okay. So your endoplasmic reticulum, ER, and Golgi complex are going to be your organelles that play central roles in the protein trafficking, essentially. The N-linked glycosylation will begin in the ER and will continue in the Golgi complex. So begin here, continue here. The O-linked glycosylation occurs only in your Golgi complex. So only here, none here. Okay? I don't know how much of that you've had before. Hopefully it fits in with what you've... Okay, dolichol phosphate. Dolichol phosphate is a specialized lipid molecule located in the ER membrane. Contains about 20 isoprene, which are C5 units. This is isoprene, by the way. This is the location where large oligosaccharides are destined for attachment to ASP residues. This is where those are going to be assembled. The terminal phosphate is going to be your end of your attachment. So this is your terminal phosphate right here. This is the dolichol phosphate. And this right here is the, there's between 15 and 19 of these. Can everybody see that okay? Okay. And that's your sort of chain. The whole thing, all of those are named alkylphosphates. And so yes, they don't have consistent formulas. They can be various lengths. Does that make sense? Did I lose anybody? Cool. Okay, so again, your Golgi complex is going to also be a sorting center. So the Golgi complex is going to have a stack of flattened membranous sacs. This is what the Golgi complex looks like if it's more like neatly organized diagram. Okay. The proteins will proceed to lysosomes, secretory granules, or plasma membrane. Don't do hot chocolate before this. Tea is the way to go. Okay, so based on signals encoded with their amino acid sequences and three-dimensional structures. These are the three different things that I was just talking about. This right here would be assembling these into a lysosome, which will then be released and release these en masse, right? And so this would be a very large release. This right here would be the secretory granule, and literally secretion, right? You're going to secrete this out. This right here is actually going to be where you take the protein and you're going to insert it in the plasma membrane. You can think of this as a lot very similar to the secretory granule, but you're not going to secrete them out. It'll just attach to the cell membrane and then unfold. And now they're in the cell membrane. And remember, cell membranes are mobile. So they don't just stay there. They're going to float around the cell membrane. Okay? Okay. Everybody knows that cell membranes are mobile, right? That like if you followed an individual lipid, it would actually wander throughout the cell membrane. Okay, so again, this is the endoplasmic reticulum ER. We're sending things off to then the Golgi. This is the cis Golgi. They'll then move up, get sorted throughout the Golgi, and this will end up in the trans Golgi, which will do the secretory granules right here, or the assembly of the lysosome right there. And then this one's the protein inserted in the plasma membrane. Any questions there so far? Okay, so specific enzymes are going to be responsible for oligosaccharide assembly. Your glycosyl transferases are going to catalyze the formation of glycosidic linkages. The activated sugar nucleotides are going to be your most common carbohydrate donor for your glycosyl transferases. These ones being the activated versions of those sugars. Okay. What you can see here is we've essentially taken, in this case it's UDP. Everybody know what UDP is? It's the uracil version, right? Okay. So UDP, the phosphates here. Okay. We've attached that then to our glucose. And notice that this is going to give us a higher energy glucose that we can then attach to other things, right? And so then we have here a something with a hydrogen on it. This is then going to have the something, whatever that was, and the hydrogen will be left on the UDP, and that will just be the normal diphosphate uracil ribose that we see all over the place, right? Specifically things like RNA and such. Okay. The blood groups are going to be based on the protein glycosylation patterns. Again, your blood groups are designated by the presence of one of the three different carbohydrates. You'll have A, B, or O that's attached to the glycoproteins and glycolipids on the surface of your red blood cells. All blood groups have a core O antigen, which is why O is the universal one, right? Because it's actually the core of everything else derivates from. So the A and B antigens are going to have one extra monosaccharide through an alpha 1,3 linkage to a galactose moiety of the O antigen. This is added by specific glycosylate transferases. The type A transferase is going to add the N acetylgalactosamine. This is going to be what forms our A antigen. The type B transferase is going to add galactose. This will form our B antigen. And remember, O antigen is the core. Okay? It's the one that gets derivated from. And this is what it looks like right here. Again, this is the O antigen right here. You can use that same key as before, by the way. It's the same ones. You also can see this in your book, and it'll have the key as well. This is the A antigen right here, and notice we've added this sugar right here, right? And then this is the B antigen, and that's where we've added that sugar. Anybody have any questions about that so far? If you're AB, what does that mean? Yeah, you have both, yeah. So you're doing both. So individuals with the O blood type will lack both enzymes. They won't do anything to that O core, and it'll just stay O core. Okay? AB blood types, they're going to express both enzymes, and so you get a mixture of the two. A will have the A transferase, B will have the B transferase. There are very important implications for blood transfusions, of course. If you feed somebody if you give them the wrong blood transfusion for the wrong blood type, it will actually cause an immune response all throughout their body and can kill them very easily. Okay? If an antigen not normally present is introduced, the immune system will recognize it as a foreign body and attack it, and it will cause havoc throughout their entire body. You know, most of the side effects, most of the symptoms we associate with things like cold, COVID, all that, those are all our immune system. There's actually very few of those things that are the actual disease. Okay? The fever, the coughing, the mucus, all of that's us. That's our immune system trying to fight it. So this is actually the cholera toxin. So our cholera, this is going to be a disease caused by the toxin from Vibrio cholerae. Individuals with the blood type O are about 8 times more likely to have severe diseases. The O antigen is going to bind more tightly to the toxin than other blood type antigens. And so this is sort of one of the reasons why we have these different blood types. Like you may go, why the hell? It makes blood transfusions really difficult, right? If we're all blood type O, that'd be way easier. But we need that variance in our antigens so that diseases can't automatically bind to it and start neutralizing our antigens, right? And then infect us and kill us, or just keep us infected forever, right? Which is like the ideal disease. That's the disease ideal. Keep us infected forever and spreading it forever. Okay? So errors in glycosylation, these can result in pathological conditions. For example, congenital disorders of glycosylation. This is going to be a pathological condition resulting from the improper modification of proteins by carbohydrates and their derivatives. The examples of that are going to be certain types of muscular dystrophy. These are going to be linked to improper glycosylation and I cell disease. So this is I cell disease. Lysosomes, these are going to be organelles that degrade and recycle damaged cellular components or endocytosous material. The I cell disease is going to be a lysosomal storage disease. Jesus, did I just say I cell a bunch of times? Oh my God. Okay, no more hot chocolate before this. L for lysosome, right? Yeah. God, sometimes I need to just stop and hear what the fuck I'm saying. I caught up though. My mouth went ahead of me, but I caught up. The affected lysosomes are going to contain undigested glucose aminoglycans and glycolipids. The active enzymes responsible for degradation are going to be synthesized. The enzymes lack appropriate glycosylation are going to be exported instead of being sequestered in lysosomes. And essentially, It's kind of like a recycling facility that's gone on the fritz, right? Instead of recycling it for the components and then assembling them into what they should be, you get a bunch of junk coming back out. Again, this is going to be the mannose-6-phosphate residue right here of your N-oligosaccharide. This again directs the enzyme from the Golgi complex to the lysosomes. In a L cell disease, the mannose is going to lack a phosphate because patients are deficient in the N-acetylglucosamine phosphotransferase. And so this right here is the lacking a phosphate. Can you see that okay? Yep. Instead of UDP, we will not attach that phosphate. Okay. Again, if you're wondering where the enzyme is in this process, here is the N-acetylglucosamine. This is the phosphotransferase right here. Okay. So that's the phosphotransferase right there. Okay, so biochemists, they use several different techniques to analyze oligosaccharide components of your glycoproteins. Your oligosaccharides, these can be detached from the protein using enzymes that cleave the oligosaccharides at specific linkages. Your mass of your oligosaccharides can be determined using MALDI-TOF. Normally, I've heard this referred to as MALDI-TOF because people don't want to have to spell it out every time. But I don't know if that's actually the official way you're supposed to do it. Write it. MALDI-TOF. They can also use mass spectrometric techniques. Many possible oligosaccharide structures have the same mass, and so you do have to be aware of that. The structures can be determined by combining additional cleavage of your oligosaccharides with mass spectrometry. You'll notice this is very similar to the amino acid method we used before. Because it is, we can do the same thing with the oligosaccharides as well, right? Though the oligosaccharides have a lot of ones where they're just isomers of each other, right? And so you have to be a lot more careful. The points of attachment then can be determined by applying the proteases to the glycoproteins and performing chromatography. I know, we just taught you that one, didn't we? Woo! Now you have that method. The fragments, they can be attached to the oligosaccharides. These have chromatographic properties that with the protease treatment, followed by mass spectrometry or direct peptide sequencing. The oligosaccharides can be sequenced by using enzymes that cleave specific glycosidic bonds. MALDI-TOF is used to identify the released sugars. Remember, that's that time of flight that we had mentioned back in, I believe, chapter 4, right? Okay. This is actually what that looks like. Again, we have the relative abundance versus your mass charge here. Okay. And we're having the mass spectrometry in this case. Note that this one right here has these units which add up to that right there, right? This one has these units. It's a little bit heavier because it has the extras. And so it has that mass right there. Does anybody have any questions about that so far? Okay, lectins. These are going to be specific carbohydrate binding proteins. So your glycan binding proteins, these will bind to specific carbohydrate structures on neighboring cell surfaces. Lectins are going to be a class of glyco-binding proteins. So again, we have the overall arcing glycan binding proteins, right? And then we have the more specific lectins. And so some of the examples of lectins are going to be mannose-6-phosphate receptor. This can bind and directs the lysosomal enzymes to the lysosome. Lectins function to facilitate cell-to-cell contact. Usually contains 2+ carbohydrate binding sites. are linked to carbohydrates by a number of weak non-covalent interactions. We have some different types. We have the C type, C for calcium requiring. The lectins, these are going to be found in animals. They're going to function in receptor-mediated endocytosis and cell-to-cell recognition. And so C-type. L-type lectins, these are rich in seeds of leguminous plants. They're going to serve as potential toxins to herbivorous insects. They're going to act as chaperones in the eukaryotic ER. The C-type lectins, that's what we're going to be looking at right down here. These are going to use the calcium ions to bind your carbohydrates. So the calcium 2+, which is right there on your lectin, is going to act as your bridge between the lectin and the sugar. Right there is the calcium 2+ in the other diagram. The two glue residues in the lectin will bind the calcium 2+. Those are the glue residues right there. You can see them over here. They're the ones that are attached to that yellow ribbon because that's your protein. Okay? Everybody see that okay? Fantastic. The other hydrogen bonds will form between the lectin side chains and the carbohydrate. And so right here, we actually see the calcium binding the mannose at the oxygens, and then the hydrogens are going to hydrogen bond the glue other oxygen right there. Okay. Can everybody see that okay? Fantastic. Okay. Selectins. I know, wonderful naming, right? Just beautiful. Selectins, this is going to be members of your C-type lectins. Notice the lectins in there. Okay. They're going to bind immune system cells to sites of injury in the inflammatory response. They'll play a role in recruiting leukocytes to the inflammatory sites. Literally, lectins that select, right? Isn't that just beautiful? There's different forms of selectins. You have the L form. This is going to bind to carbohydrates at the lymph node vessels. You have the E form. This will bind the carbohydrates to the endothelium. And the P form. This will bind the carbohydrates to the activated blood platelets. Notice these letters actually are, again, lymph for L, endothelium for E, and then P is platelet, right? For blood platelet. Okay? Somebody really thought when they did that naming. It's such a good one. They're not always good, right? But when they are, gotta appreciate it, okay? A little smile. The influenza virus, anybody knows influenza, right? Okay. This is going to bind to sialic acid residues. Hemagglutinin is the influenza virus, is going to be, that's lectin protein. They'll bind to the carbohydrates that are the sialic acid residues that are linked to the galactose residues on your cell surface glycoproteins. The virus is going to be engulfed after binding. The virus will then replicate inside the cell, and viral particles will bud off of the cell. The assembled viral particles are going to be attached to the sialic acid residues of your cell membrane. This is done by hemagglutinin, neuroaminidase, or sialidase, which is by far and away the easier naming for that. By the way, what does it act on? Sialic. Sialidase. That's the influenza virus protein that's going to cleave your glycosidic linkages between sialic acid and the rest of your glycoprotein. This is actually what frees the virus to infect new cells. Otherwise, they would just be budded off your cell, and you'd have this cell with a ton of influenza trailing off of it, like a shag coat. Okay. The inhibitors of the neuroamidase, Tamiflu and Relenza. These are actually important anti-influenza agents. This right here is what we were talking about in a wonderful, beautiful diagram graphic format. So again, we have our virus. It's going to use the lectin for the specific cell binding. This right here is your hemagglutinin, right there. This right here is the endocytosis. And so we're going from the outside, we're binding to this target cell, then entering into the cytoplasm. Okay? This right here is the assembly and then budding. And so you're going to assemble and bud through here. This will then have a full virion particle that is virus that is attached to your cell membrane. And so then you're going to have your release. That's that neuraminidase that we had been talking about before. Anybody have any questions for that? No? Okay, yeah. I always kind of wonder if when I start using more COVID examples, will I get my YouTube channel canceled?